The broadcast is now starting. All attendees are in listen-only mode.
Hello, everyone, and thank you for joining us today. You've logged on to the Nelson Labs webinar, Appropriate Uses for Product Sterility Testing. When is sterility testing appropriate? Just a few housekeeping items I'd like to take care of before we get started. If you miss any of our webinars or would like to refer back to them, you can always find them on the Nelson Labs website on the on-demand webinars page listed under Education. You can receive notifications for upcoming live and on-demand webinars by following us on Twitter, LinkedIn, or on Facebook. We also invite you to register for free for the Sotera Health Academy at soterahealth.com/academy. There you will find curated content from industry thought leaders, from introductory sterilization and lab testing topics to advanced in-depth learnings. Our expert advisors have filled this academy with cutting-edge educational content and resources to help you mitigate risk, go to market faster, and achieve excellence in your field. Nelson Labs is a leading global provider of laboratory testing and expert advisory services for medtech and pharmaceutical companies. The company performs over 800 rigorous microbiological and analytical laboratory tests across the medical device, pharmaceutical, and protective barrier industries. The experts at Nelson know every test matters and requires solutions to complex problems to improve patient outcomes and minimize client risk. Let's get started. Today we're joined by two excellent speakers in Bryce Telford and Jennifer Gygi. Jennifer has worked over 25 years in the medical device laboratory field. She has worked in the microbiology, bioburden, identifications, and packaging sections at Nelson Labs. Jennifer has experience in various microbiological tests, or microbial tests, including microbial limits, plate counts, biological indicator, population verification, organism identification, and bioburden tests. Jen has spent the past 15 years in our packaging group and is now as a part of our expert advisory services. As one of the original packaging group members at Nelson Labs, Jennifer is heavily involved in validating all the packaging test methods and equipment and writing the test procedures for those tests. Bryce is a recognized authority and industry leader in radiation sterilization, bioburden, microbiology, and tissue processing. He has over 14 years of experience working in laboratory functions in research, test design, media formulation, medical device, and tissue industries. He works with clients in all areas of the world to speak, train, and consult on failure investigations, bioburden issues, establishing bioburden alert and action levels, product family grouping, product adoption, radiation sterilization, and tissue validations. We encourage your questions. We will try to get as many as we can at the end of this presentation if there's any extra time. And if we can't get to them, the questions will be passed along to Bryce and Jen. So now we'll turn the time over for today's presentation to Jennifer, who will start us off.
Thanks, Mike. As he said, my name is Jennifer Gygi. I'm joined here with my associate, Bryce Telford. We both work in the Expert Advisory Services group here at Nelson Labs. And we decided to do this particular webinar because we've been getting a lot of interesting test requests and questions around sterility testing and when it's appropriate to use it. So hopefully this will be a good educational experience for us all. And let's get right into it. All right. Let's figure out how to make it work. Okay.
So when we talk about sterility testing and we think about it, there's kind of the old mentality and the way that sterility testing kind of started. And that's really using sterility testing for your batch release testing. So we have a product, we make it, we sterilize it, so we're going to do sterility testing on it in order to do that batch release testing and to show that it's sterile. However, over time, that's really evolved, and the newer way that we really handle sterility testing is that it's part of a validation process. It's used for a monitoring, or we're using it to assess a change to the product, so it's part of our change control process. So concepts for sterilization is that really the bulk of the work that we do when we do sterility testing and we look at it is it's going to be for validation purposes. So it's being used to show through a validation that a particular SAL or sterility assurance level has been achieved. Then once we've validated our product and our sterilization process, and we have the sterility testing that goes with that validation, we're then going to use it as prescribed by our various standards or pharmacopoeia for monitoring purposes. So we do the test of sterility to monitor product and to ensure that it is compliant to what we're trying to do. Finally is going to be our change control. So if I make a change to the product, I want to ensure that that change does not affect the sterilization or the safety of the product. Now, you'll notice that that really isn't going to occur very often. It's a small part of this pyramid, and it's only applicable in certain key situations that we're actually going to do sterility testing as part of our change control, or use that sterility test to show acceptance of the change. So let's kind of take a look at that process and take a look at how that works. So the sterilization validation, typically when we're talking about industrial sterilization, we're talking about ethylene oxide sterilization or radiation sterilization. So we have the applicable standards for that. So we have our ISO standards that prescribe the sterility testing and the sterilization that we're going to be doing. So for EO, that's going to be an overkill or a BI bioburden-based method. For radiation sterilization, it's all bioburden-based, but we have a few ways that we go about doing that validation, whether it's BD-MAX, method 1, or method 2. And then once we've completed that validation step, then our monitoring is going to be prescribed by that set ISO standard. So for EO, that's going to be our biological indicator, our PCV testing, things of that nature, where we're doing our release or our parametric release parameters. For radiation, monitoring looks like your dose audits. So previously, and kind of common in the industry, we would refer to those as quarterly dose audits. We're kind of trying to get away from that name, quarterly dose audits, as there's some misunderstanding there. But the long and the short of it is that we're using the test of sterility as part of the monitoring prescribed by the standard. And occasionally then, as with any product, especially with global supply chain issues, with supply constraints, with personnel constraints, there needs to be changes to your manufacturing, whether that's changes to raw materials or changes to the process. So that inevitably leads to change control. Both standards give guidance on how to handle that change control, but that change control is going to be per your initial procedures, and it's going to kind of prescribe how we're going to use our testing and what testing may be really appropriate for that. So whenever we talk about sterilization, what we're really talking about is usually terminal sterilization. And we also are going to touch on the pharmacopoeias and what that means. But for the most part, when we say sterility, what we're actually talking about is a sterility assurance level. Now, nobody likes saying sterility assurance level over and over again, so we refer to it as your SAL. And what that really is your probability of an organism surviving the sterilization process. So for a typical product to be considered sterile, the industry standard and the standards themselves, indicate a sterility assurance level of 10 to the minus 6. That means there's a 1 in 1 million chance of something surviving the sterilization process. So let's say I have something with my product and I test 10 samples. what is that 10 samples actually showing? Well, that's a sterility assurance level of 10 to the minus 1 or a 1 in 10 chance. I'm only to an SAL of 10 to the minus 1. If I take it a step further, if I test 100 samples, I'm still only demonstrating a sterility assurance level and SAL of 10 to the minus 2. So that brings the question, what's needed to demonstrate for terminally sterilized product for it to be considered sterile? So let's say I have a product, it's received its full exposure for EO, it's received its full dose for radiation. What do I have to do to actually demonstrate if I take those products to show that it's sterile? Well, the answer is you have to test 1 million products. Now, is that going to be something that's reasonable and practical? Nobody's going to really sterilize 1 million products, test 1 million products in order to show that, yes, I achieved sterility with this. So that's where the validations really come in, is you're using things such as fractional cycles, half cycles, verification doses at the lower SALs, and then that's correlating to your sterilization. If I want to take a product that's been fully exposed and I want to show that it's sterile, I better be prepared to test 1 million of those. Colloquially, for fun, we figured out with our Salt Lake City clean rooms here at Nelson Laboratories, how long would it take to test 1 million samples? So we figured if we used every one of our clean rooms, if we had 2 shifts, a morning and an evening shift, and we ran 5 days a week, it would take us 2.5 years to test 1 million samples. So are we really going to test exposed product to show an SAL of 10 to the minus 6? No. And that's what we need to consider when we're talking about doing a test of sterility. Are we really showing what we think we are when we do that test? Are we really demonstrating what we want that test to do? If we only test 10, remember, we're only at an SAL of 10 to the minus 1. To be considered sterile, we need that 1 in 1 million, that 10 to the minus 6. So that kind of leads to some of the questions that we've received over the last few months and over the last year of, well, I have sterile product, and what do I do with it to show that it's still sterile or to extend its life, to basically assess that sterility? So that's where we get to the next part.
All right, so as Bryce is saying, we've got our sterilization validation. That's going to prove that whatever sterilization cycle that we're using is going to provide that desired SAL of 10 to the minus 6. But as soon as you bring it out of a sterilization chamber, it's not going to remain sterile unless you have some kind of a protective barrier, and that is where packaging comes in. So this is really a kind of a 2-part. We have multiple validations. There's the sterilization validation, the packaging validation. The packaging validation is going to prove that the sterility can be maintained, that sterile condition, over time. And we're going to do that by testing the packaging in a new condition after we've done some sterilization and distribution processes with it. And then we're going to test it again at the end of its life to show that it is still performing as expected. And of course, this validation isn't just one simple test. It's based on strength, integrity, and microbial barrier properties of that package over time.
So as we like to say, there is no sterile product without a sterile barrier to maintain it.
Right. And just like we have the pyramid for sterilization, it's the same concepts when we're talking about packaging. We're going to run these validations, and that forms the foundation. And that validation proves that we can do things the same every time. We're going to monitor the validated processes to make sure that nothing has unexpectedly changed, that my output is still the same. And then if I intentionally want to make any changes, I have change control processes in place to evaluate and decide what kind of additional testing may be needed to make sure that change is not going to have a negative impact. But when we're talking about a packaging validation, again, it's in layers. There's multiple stages. This here is an example of a sterile barrier pouch, the most common of which would be maybe a Tyvek film pouch. You've got two materials there. You've got a Tyvek, you've got a film. Both of those are going to be produced by a supplier in a large roll stock. Those suppliers are going to have performed equipment validations to show that their equipment operates as it's supposed to. They're going to have some kind of a process validation to show that each roll that comes out the back end of that machine is identical, that it's the same, and that it meets whatever that acceptance criteria is. They're going to have some kind of monitoring in place to make sure. They'll periodically pull sample and test it for certain properties. They may have inline monitoring that may be doing some kind of inspection or thickness or something to make sure that everything is within specification. And then they're going to have internal change procedures in case they need to make a repair to a piece of equipment or they need to change supplier. They've got procedures for that. Then it's going to go to the next stage where it's going to go to a converter who's going to take this roll stock and make it into a preformed sterile barrier. They're going to have specialized equipment that would be validated, that's going to take that roll stock, size it into these packages, and they're going to have sealing equipment that's going to put the seals on these packages. All of that's controlled by a validation. They have monitoring in place to make sure that their process is the same and it's under control, and then they have change control procedures in place for any changes they need to make. Now they'll sell that preformed sterile barrier system to a medical device manufacturer who will add that final seal. That's your packaging process validation. Again, you perform the validation, you use monitoring to make sure that everything stays within the scope of the validation, and then you have change control for any changes that you need to make. And then the final step here is our final packaging system validation, which is the one that I was talking about earlier, where you would take your product after sterilization, after distribution, you would test it in that new and that old condition. Again, you perform the validation, you have some kind of monitoring that you may be doing to make sure that your output of your process is the same every time, and then you've got change control procedures in place. So it isn't just a one test, it's multiple things building up these layers. And how do we really know how to perform validations? Those last two validations are going to be in ISO 11607. So that's going to cover your process validation and then your sterile barrier stability piece of your validation. And that particular standard is talking about strength testing, right? That's a physical test. Integrity testing, those are physical tests. Microbial barrier, yeah, it sounds like it might be a sterility test, but when you actually read it, it's a physical, more of a physical test. You're using microbes. If it's a porous membrane, you're using microbes at a high flow to try and push them through the porous membrane and see how many come out the end. Or if it's a non-porous barrier, you would be doing the same type of a test, only it's with air. Can I get air to go through the material? If not, it meets this definition for non-porous. The same thing is going to be in the pharma world. We've got a document from FDA saying container closure or system integrity testing is preferred over sterility testing for those stability tests of, say, vials or syringes. And that is really just pharmaceutical packaging. So our standards are preferring the physical testing over sterility. Why would this be? Well, there's three reasons. First reason is that it's faster. I can run a bubble test on a set of samples in an hour and I'm done. If I want to run that same set of samples for sterility, maybe it takes me a couple of hours, but then I have a 14-day incubation before my test is done. The second one would be sensitivity. Microbial testing is nowhere near as sensitive as physical methods. And that sensitivity becomes really important when you're looking for small defects. And the third one would be contamination. Physical tests don't have an element of contamination. Microbial tests are always subject to that possibility that you may get erroneous data due to contamination. And so that's why we really prefer to do physical testing over sterility. We've got an article here, the third bullet point. This is an article that was put out in 2018 And it's got a really catchy phrase, to test or not to test. That is the question. It's full of great information, very well written by a panel of industry experts. So you don't have to take my word or Bryce's word that this is the way it should be. Again, you can go and reference this article. And we often will give this to people who are asking these types of questions so that they could go read this. And it usually is pretty convincing to them as to what we're about to tell you right now. I put some excerpts from the article. This is just a teeny weeny sampling. So kind of wet your whistle as to what kind of information is in there. The first piece here is table 2. And this is just a comparison of sample size to demonstrated SAL, right in line with what Bryce was saying earlier. If you only test 10 samples, gives you an SAL of 10 to the minus 1. And you're thinking, well, I've increased my sample size up to 30. Yeah, that's going to still only get you an SAL of 10 to the minus 1.5 or so. So if you think that, you can do a sterility test on a small number of samples and have any kind of meaningful claim of sterility, it is just not there. This second piece here is really going to illustrate why we prefer those physical methods. In this, they've quoted a test that was done quite a bit previous. It looks like it was published in 1995 in MDDI. And it looks like it's an aerosol challenge type test where they took whole packages, they put defects in some of them of varying sizes, and they exposed them to an aerosolized spore in an exposure chamber, and then they did a sterility test on the packages to pull what was inside out and tested it. The results are, they're pretty interesting here. I have group A, that's 30 samples with no defects. These are perfect packages, and these should be awesome, yet I have one positive. Now I'm going to assume that that positive is a false positive because these packages are beautiful, so why in the world would they have a growth? Yeah, that's going to illustrate my problem with contamination. 1 out of 30, that's probably a contaminant, yet I will be very hard pressed in failure investigation to conclusively prove where that organism came from. Very difficult.
And when you're working with the standards, when you're talking sterilization, I test 30 samples, I have one positive. It should be fully sterilized product, and yet I have a positive. So what do I do now? Well, that's why it's important and critical to consider your sterility testing, because what do I do? There's no ISO standard telling me what my acceptance criteria or how to handle that is. I've tested 30 samples, nothing really guides me on that. They should have been fully sterilized. I have a positive. Well, what does that positive mean? And this is an example where that is exactly kind of what happened. And let's look at some of the other sizes.
Just going back to that really quickly, let's say that I had some old product. I wanted to test it to prove that it was still sterile.
Yeah, I want to show that my sterile product's sterile.
Yeah, it's been sitting in my warehouse. It's past expiration. I test 30 of them for sterility. I get one positive. Not only does that say I can't extend my shelf life, but what does that say about all the product that I've been putting out for years under this validation?
Yeah, it calls things into question. It raises a lot of concerns of, well, is my packaging and is my sterility, is the overall safety of my product ensured when I have something like this show up?
So that's the contamination piece. Let's look at the other critical component of why we would prefer a physical test, and that is sensitivity. You can see here that I have these 3 different defect sizes. So that's 90 samples with holes in them. Put in a chamber with a high microbial load, then tested for sterility. And of those 90 samples, I have 2. So that's 88 samples that should have failed the test because the packages were defective, yet they passed this test. And that really shows us the sensitivity problems. I can tell you that if I ran those 90 samples, because that smallest defect that's in there is actually the sensitivity for bubble. And I know I can get at least 80% of that defect size with the bubble method. I would be expecting to see high 90s % of recovery in a physical test. Big difference there in the sensitivity. All right. We realize that it's hard for some people who haven't ever really seen what some of these tests look like to understand how I could get contamination, or maybe why I wouldn't pick up a failure in a test on a package with a hole. So we've got a video here. This is on YouTube, and it's one that we put out that shows a sterility test yeah, let's get it going. It shows the sterility test. Whoops.
Technical difficulty.
Yeah. All right, here we go. Now you'll notice that we take precautions.
It says it cannot play media.
It cannot play the media. Oh.
Our apologies, everyone. So the video is available on YouTube. Our apologies that it's not working today. It worked in
our demo.
Our practice session, but our apologies for that. But the long and the short of it, if you can visualize it, feel free to go to YouTube later and you can see the video.
Just type in Nelson Labs sterility and it'll pull right up.
A sterility test is not some magical thing. What we basically do is we take a device and we, as aseptically as possible, put that device into growth media. So our analysts wear a clean room gown, they're covered from head to toe, they have safety goggles, they have masks, so that there's no skin exposed or anything, in order to show that as best as possible, any organisms are coming from what's being tested rather than the testing system. However, there's still handling, there's still manipulation, so there's still those chances for contamination on that test of sterility. And if I have a very complex product, I'm doing an awful lot of manipulation and an awful lot of handling.
Right, and where's the last place that an organism is going to, the hardest to sterilize spot? It's going to be the inside of the device.
Right.
So we have to cut the devices up so that we can get all those interior surfaces exposed as well. That manipulation does open up a lot more possibilities for contamination.
So let's talk about test of sterility, test for sterility. So terminology is something that really kind of matters within the industry. So the breakdown of a sterility test is that test for and test of. So what's a test for sterility? Well, that's defined by pharmacopoeia. So there is harmonization between, for example, USP, European Pharmacopoeia. For the purposes of this presentation, we're going off of what USP specifically says. And what we do is we do a test for sterility, and we highlighted some key parts here from the Pharmacopoeia. So what we have is a satisfactory result only indicates that no contaminating microorganism has been found for that in the sample examined under the conditions of the test. That means that specific sample you're testing is considered sterile, and only that specific sample.
And only in the conditions of test.
And only in the conditions of the test, the way you set it up, because how you run a test is going to affect the outcome.
Right, what kind of media you use.
Exactly. So this is not demonstrating a sterility assurance level. There's no claim of what this means for other products and things such as that. This doesn't demonstrate a sterility assurance level, and it's intended specifically for pharmaceuticals and aseptic processings. And it's noted within that USP that this is not intended or designed to ensure a batch of product is sterile. That's part of the validation that you're doing. It says that the validation is part of it. The test for sterility is not designed to ensure that a batch of product is sterile or has been sterilized properly. So a way to think of the test for sterility and what USP is prescribing is it is a monitoring step. It's a USP prescribed monitoring step Similar in concepts to the test of sterility for your dose audits or for your BI testing. It itself is not intended to demonstrate that the product's sterile.
So maybe you should define aseptic processing versus terminal sterilization.
That's a very good point. So aseptic processing, also sometimes called sterile processing, is I'm relying on everything I'm bringing into the system being sterile already. So chances are it's been sterilized in one way or another. So let's say I have a vial with a pharmaceutical in it. Well, the vial has been terminally sterilized. And we'll say what that is in a second. But then I have the pharmaceutical. That's usually going to have gone through some sterilization process of its own, such as a filtration sterilization or maybe some form of heat sterilization. But what is happening is I'm taking these two then sterile products and I'm combining them together in a sterile way to maintain that sterility. So the sterility is being maintained rather than being performed. Terminal sterilization, quite simply, it's terminal. It's at the end. Well, what does that mean? I'm sterilizing the finished product in its sterile barrier system, in its packaging. So terminal sterilization, it's done at the end. Aseptic processing, I have steps leading into it that have to be sterile, and I'm maintaining that sterility. So what's a test of sterility? Well, that's really intended for your terminal sterilization. So it's part of demonstrating your sterility assurance level, and we have some definitions of a test of sterility from the ISO standards. But one of the key things that's pointed out in the ISO 11737-2 series, which is the standard when it comes to ISO sterility testing for terminally sterilized product, is it's noted that if I take product, and it says right there, it should have very low probability of microorganisms, such as 1 in a million, and it's noted right in the standard that if I test that for sterility, it really provides no scientifically usable data, and it's not recommended. I'm not demonstrating sterility by doing this test. That's done through the other validation steps that correspond to a verification dose or a half-cycle fractional cycle. So testing fully sterile product, no scientifically usable data really comes out of it. So when we do a test of sterility, it's for product that's terminally sterilized, and it's really intended to be on product that is verification dosed, fractional cycled, your BIs, things of that nature, not fully exposed, fully dosed product.
Okay, let's look at some case studies. We've created these case studies based on the types of questions that we typically see. So we'll go through them together. The first one here is a partially sterilized product. So these samples were processed through the validated sterilization process. You ran them through your EO or your radiation, and you're reviewing the data, and you notice that the load's either underdosed if it's radiation, or the cycle, something went weird in the cycle for EO. How about, can we perform a sterility test on some of that product to ensure that the load's sterile?
And this is very timely. This is something that unfortunately is out in the industry right now due to some issues when it comes to radiation dosing that have come up. But this is also where your overall validation is critical. And it's why you need to think of the validation ahead of time. And if you haven't, now's a good time to pause and do that. Because let's say I have a product that has been validated via ethylene oxide. Well, as part of my packaging validation, as part of my biocompatibility, as part of my other validation activities, I should probably validate a 2x sterilization process or cycle so that if there is an issue with the exposure, if there is an issue with the load, then I can go through and I can send it through again. It didn't get its full cycle, so let's just run the cycle entirely over again. So the second part of that is radiation. Well, fortunately, radiation dose is considered cumulative. So this is where your max dose validation is important. I want to be able to give myself some flexibility that if there's an issue with my load, then potentially I can go back and I can do what we refer to as topping off. So if there's an issue with the dose, I can send it to get more radiation dose. But the key here is, can I just do some sterility testing on product from that load and show that it's still sterile? Well, we have to demonstrate that SAL, and that's going to be a 10 to the minus 6. Chances are, unless it's a high volume product, such as an adhesive bandage or swabs or something like that, I don't have 1 million products. And then also with that, I don't have 2.5 years for those 1 million products to be tested by the laboratory to release that batch. So if there's an issue like this, sterility testing really isn't the answer. It's how was my product validated? What options do I have to get to that sterility assurance? And again, this is why that 2x sterilization is important for EO, and that's why your max dose is very important for your radiation sterilization.
Yeah, so if you don't have that data, you either have to get it after the fact, which nobody wants to do, or you just have to scrap the load.
And nobody wants to scrap a load.
Nobody wants to scrap a load. All right, let's look at case study 2. This is an aseptic process validation. So again, that's the one where we've got sterile vials, and then we're going to put a sterile solution in, and we're going to cap it in a very clean environment and hope that that we get sterile product on the back end. Right, so we're using an aseptic process to assemble previously sterilized components in this clean environment. How about using a sterility test to determine if the final finished form is sterile?
So if the final finished form is sterile is kind of the key. There should be validations that lead into this that are showing, so we have the picture here, we have the vials. Those vials going in should be sterile, and they should have a sterilization validation with them. Whatever we're putting into them, chances are in this case for a filling, it's going to be something that may be sterile filtered or something like that. There's a validation for that. So what we're doing and what we're validating ahead of time is our sterilization validations. So what we're doing after on the finished product is basically a monitoring step to show that when I made all these sterile products and I brought them together, did I keep that sterility? So am I able to take that finished product and show that it's sterile? Well, no, because there's no SAL being demonstrated for aseptic processing on that finished product. It's all the other validations leading into it. It's all the work going ahead of time into that. So am I going to do sterility testing? Yes, that's part of what USP prescribes.
Right. So,
but I follow what USP says, and I have those other validations with it. And the key is that final validation on that final product isn't showing my SAL. It's not showing that claim of sterility.
no, but I would probably say that, because we haven't talked about the filling process validation yet. You did mention that all the components would need validations to show that they were achieving sterility that way. But just to kind of give you an idea, because we have seen a few of these, when you're doing a process filling, aseptic filling validation, again, you're not looking at sample sizes in the 10 to 100. You're looking at sample sizes 1,000 plus.
And hundreds of thousands even.
Sometimes, yeah. And then instead of filling it with your drug product, you would fill it with growth media and then just incubate all those. So you would be doing, sorry, real high volume. All right, let's look at another one.
So this is one, you're trying to establish a shelf life for your product. So I have a product, I'm trying to bring it to market, and I want to come up with my expiration date. I want to do my shelf life testing. I'm probably going to be doing my accelerated aging testing with it. So with that accelerated aging testing and with that shelf life testing, am I going to pull those sterile products or those old products off a shelf? And can I sterility test those to show that, hey, I still have sterile product after this expiration date?
Well, you won't be surprised by this answer, but you don't have enough samples to reach the desired SAL. So the way that you would do this is to perform a packaging validation If you did your packaging validation originally with a shelf life of 1 year, and now it's been almost 2 years, and you've got some old products sitting on a warehouse shelf, you don't run a sterility test on those. You go through and you take the 2-year-old product and you perform another time point of that packaging validation, where you would run a strength, an integrity, microbial barrier on it, show that all those properties are the same. And again, those are physical tests showing that even now that it's a year older, those materials and those seals and the package is all just as good at being a sterile barrier as it was at the 1-year time point that you've previously done.
And I think that speaks to some of the sterility testing and stuff so far is, let's consider if I've demonstrated and I've validated my product sterile, it's not going to just become unsterile. An organism's not going to come into existence. As one of my associates, it's a bit blunt, but points out, we've disproved spontaneous generation. So if it's sterile, and I've shown it's sterile, then I'm not going to suddenly have that organism show up. So where I'm going to get that organism is I'm going to have it in a failure of my sterile barrier system, my packaging.
Yeah,
that's going to be
those are event related.
Yeah, something's going to happen to cause me to lose sterility. That means I have to have a failure of that barrier. So that's where this packaging testing comes in.
Yeah. And speaking of some weird ones, let's talk about something abnormal happening during storage. Because again, you wouldn't think that that storage could be, it's just sitting on a shelf. You know, what bad things can happen just sitting on a shelf? But we have seen a few instances where there's an unusual event that happens and you don't know if it's compromised the packaging. Let's say it was exposed to, you know, really high temperature, really low temperature. Maybe it was exposed to a lot of moisture for one reason or another. How about doing a sterility test here on those packages that's been through this event? They may be affected, and if we sterility test them, then we'll know if that event damaged them.
And this is something that is timely. Natural disasters happen. People have issues with their products. And this also kind of assesses the entire validation as a whole. You have to consider all of the validation of your product. And what is a sterility test really showing? So let's go use 2 examples for this particular case study. So let's do the first one, which is I have a humidity event or I have a water event. A hurricane happened, unfortunately, and some of my boxes got wet or something like that. Can I sterility test it? And if I pass sterility, does that mean my product's okay? Well, first of all, I'm kind of relying on those microorganisms. So if my packaging is intact, just like we discussed with the other slide, I shouldn't lose sterility. So packaging testing probably going to be more applicable than sterility testing. But even if I have a sterility test pass and my products got wet, that doesn't mean my product's okay. So Tyvek is essentially a really good filter. So if I have a product that got wet, I might not have microorganisms crossing that filter, but maybe I have other things that I don't want. If I have rain and it's falling off the roof, if it's going through the roof, it's picking up every gross, disgusting chemical that's coming along with that, and that may be able to cross
well, even if it doesn't, if it's on the outside of your package.
Right. Do I want to wipe that down? Is that something I really want getting processed into a hospital? So I have to look at the entire situation. Is a sterility test really showing my product's okay? No. It might still be sterile, but one of the sayings we have is clean doesn't mean sterile, and sterile doesn't mean clean. So I might have sterility, but that might not be what would be considered a clean product. I have some form of contaminant somewhere. But let's say also, and this gets to you, Jen, in the packaging, well, what happens if it's summertime and my package sits on an aircraft for an extended period of time, or on a dock at the airport, or it's wintertime right now? What happens if it sits in the cold and I'm not able to ship it due to flight delays or things of that nature? What do I do with those anomalies?
Well, I'd say that first of all, you don't have enough samples. This is a recurring thing, right? You don't have enough samples to reach the desired SAL, so just testing a few samples for sterility won't give you any meaningful data. But these are actually one of the funner things that I get to do is these kind of assessments, because you really get to kind of drill down and figure out what that event was, what kind of potential impact that it could have on not just the sterile barrier, but the protective packaging as well. And really most of the time when you get through these, there's some kind of physical test that you can do in conjunction with this risk assessment that you're developing over, how likely is it that that event damaged things? What can I do to do some kind of physical test to show that my barriers are still intact? And so that's usually the better route to go.
And at the end of the day, we can consider sterility, we can consider packaging, we still have to consider everything else. With that temperature excursion, is my product still okay? What if I have a combination product and it's a pharmaceutical? Well, I need to assess that too. A sterility test isn't going to show that. It'll show sterility. Okay, so I have 10 samples. I've shown an SAL of 10 to the -1. Well, I still haven't shown sterility, but I also still haven't shown my product's still okay.
Right, and that's something else you would be looking at in a risk assessment, is not just from a sterility assurance level, but how functional, did that event damage the product in any way as well. So let's move on to case study number 5. This is a lot release. So you're setting up a lot release or a monitoring program. Should sterility testing be part of that?
So this is one of those times where the answer is, and it's one of our, for anyone that's ever worked with or interacted with Nelson Labs, one of our favorite answers is, well, it depends. So if I'm doing lot release testing or monitoring programs, should I do sterility testing? Well, the answer is, of course, terminal sterilization, no. I'm going to do a test that is on the verification dose product. So is sterility testing going to be involved? Yes. Is sterility testing on terminally sterilized product going to be involved? No, as we see on the screen. Well, if it's aseptic processing, if it's a pharmaceutical, yes, I am doing sterility testing on that in accordance with the United States Pharmacopoeia or my applicable national pharmacopoeia, and I am going to have sterility testing as part of that. So there are certainly places where sterility testing, yes, I'm going to do it. Yes, it is appropriate.
Well, I got a wrench to throw in here for you. Let's say you've got a notified body and you're trying to do your submission and you're following the standards, you're doing what you think you should. You send this file away and the notified body says that they want to see a sterility test.
So this happens. So we have combination products. So I have a product, it's terminally sterilized. I have the product, it gets the drug coating, it's put in its final packaging system, and it's terminally sterilized. So what does that mean now? Because the auditing body wants to see, because it's a drug component, they want to see USP, but it's also terminally sterilized, so we want to follow ISO in our validation. So that gets to be a bit of a gray area, and that's one of my favorite quotes that I actually picked up from a client that we were interacting with, and that is, the wheels of bureaucracy turn slowly. The reality is, if a notifying body wants it, sometimes it's just going to be faster to do a 14-day sterility test than it is to try and escalate that and work it through the auditing bodies on whether it's appropriate or not, what testing we should do. But there's always that caveat when it comes to doing this testing, and consideration of why you should potentially push back is, as we showed with that previous testing, test 30 samples, had absolutely no defects, still had a sterility positive. So what if I'm appeasing a notifying body, I'm taking sterile product, and I'm testing it to demonstrate sterility? Well, what happens when I have that positive test to sterility result? I don't have guidance from the ISO standard because I'm not following the ISO standard, for example, on verification dosing or half cycles. Instead, I'm testing terminally sterilized product. So I've left where the guidance is. So it's kind of a case of the notifying body may ask for it. You may feel inclined to just go ahead and do it to appease them, but there needs to be that consideration. Well, what happens and what does it mean? And if you have that positive and you try to justify things after the fact, well, then it just sounds like you're rationalizing, oh, my product's okay for these reasons, or, oh, it's still terminally sterilized in that pass, so it should be acceptable. So if you're going to go through that, have guidance on what things mean ahead of time. And it's kind of a sad situation. We've seen it before where people do this to appease the auditing body, and they do have a positive test of sterility. So then it leads to a whole investigation and looking at the lab. And chances are fairly reasonable that, and we saw it with the 30 samples tested, 1 positive, lab contamination does happen. But trying to just pin product safety on lab contamination really doesn't demonstrate improved product safety. So in a lot of situations, if you have that positive, it might mean you've thrown away the whole batch for something that isn't necessarily a requirement to do.
So what you're really saying is, it's up to you. It's up to you if you want to just run the tests to get it through. We're not going to judge.
Not going to judge. We'll go ahead and do the testing, but be aware of what that means and be aware there isn't really guidance for you if there is an issue. If you have a positive and you call us up and you go, what do we do now? Well, no standard really says.
All right. And with that, let's look at our final thoughts. So kind of to review what we've talked about. First one, sterility testing is not appropriate for terminally sterilized product, but it can be. It's part of your validation testing, but all those are done on partially sterilized, not terminally, not full sterilization, but partially sterilized.
Yes, your fractional loads, your half cycles, your verification dosing. So sterility testing may be used for monitoring. Again, it's part of USP if you're doing aseptic or pharmaceuticals, but as it says, only applies to those samples tested under the specific conditions of the test. It does not say anything about the lot actually being sterile.
Yeah, well, physical test methods are preferred over sterility tests because they are more sensitive, they're faster, and they're less prone to contamination or false positives.
That's the key. Are we having a true positive or do we have a false positive? And
it's really hard to get a conclusive determination in a failure investigation. It really, really is.
So that's really what it comes down to, is it's best to follow the standards. So one of the sayings that we have around here, I've heard it from a few people, so I don't know the original quote. I'll give Dr. Jerry Nelson credit today, is the most conservative approach is to follow the standard. There's times where people will be, well, I want to do this because I feel it's more conservative, or this is a worst case. Well, eventually you can make things such a worst case that you've made it basically impossible to pass. Or you've decided, oh, I'm going to do this because I feel it's more conservative, but now you've left the guidance and standards. So there's really no acceptance criteria or guidance on what to do if you fail. So the most conservative approach, follow the standards.
All right, and here's a fun one. Product is considered unsafe until it's proven otherwise. So when you do have a false positive, now you've got to prove that was a contaminant and not a failure of your entire sterilization process.
And this is really what things come down to at the end of the day. We're ensuring patient safety. And when it comes to patient safety, your product is guilty until proven innocent. It's unsafe until it's shown to be safe. You have to have the data showing it's safe and acceptable so that it's safe for a patient.
Okay, here's just a review about our Sotera Health Academy that was mentioned by Mike at the beginning. And now it looks like we may have just a couple minutes for a question or two. So we turn it back to you, Mike. We got any questions in the queue?
Yes, thank you, Bryce and Jen, for all of this information and your insights. We do have time for a couple of questions. So I'll start off here with the first question. And that one is, can you discuss the difference between sterility and stability?
Okay, so sterility is pretty much the absence of microorganisms, right? That's
the
what
ISO definition.
that's what we're talking about, that 1 in a million probability of having a non-sterile unit after sterilization. So that's sterility. Stability is how something performs over time. So usually stability is an aging study. We'll run some kind of testing at a baseline, and then we'll age it, and we'll run it again after aging, and we'll show that it's the same. We do stability with packaging. They call it in the pharmaceutical world, they call it stability testing, which is technically on the packaging and also on the product. We don't use the term as much with device testing stability, but the concept is the same, is that we're showing that over time that nothing has changed significantly.
So an example for a medical device might be something like your biocompatibility. So if I have a product, let's say it's an implant, is it stable over time, or am I seeing increased leachables, decreased leachables, something like that? But really it comes down to, is my product stable in its condition, or is it free of microorganisms?
Great. Thank you for that response. We'll take one more question. And that question, it's a rather lengthy question, but I think it sounds important. So the regulatory authority in China is interpreting the FDA guidance on stability testing in lieu of sterility tests to say that we are required to perform tests on package integrity at the end of shelf life for every batch, or minimally at least one time per year. This is for the 510K device. They said they haven't heard of anyone doing this before, and if a packaging system is validated per ISO 11607, does this really need to be repeated for verification of the SBS stability? What's your interpretation, guys?
So I'm going to assume here that they probably have a flexible barrier package that they validated per ISO 11607, which will go back to that. The most conservative thing you can do is to follow the standard. They've done what they should do. You follow the packaging standard to do a packaging validation. And I don't know unless, because container closure integrity testing is normally done on pharmaceuticals, and they're normally rigid containers. They have been able to adapt some of the methods to flexible barrier packages, but they do definitely work easier and better on rigid packaging. So I'll just assume that that's a flexible barrier, that they followed the right guidance for doing that. And this is one of those instances where you have a notified body that's asking for something weird because they're interpreting the standard differently than the rest of the world. And then you get into that rock and a hard place that we were talking about a little bit earlier, where you can appease the notified body by giving them what they want, or you can try to push back. Sometimes pushing back is kind of futile. And so you're just kind of stuck in trying to decide what the best course of action is. I'm interpreting it as, if you follow the packaging standard and you've performed your packaging validation for the packaging standard, then that should be sufficient.
And if it's something that somebody is specifically concerned about, that's guidance and responses to auditing bodies is something that expert advisory services does. So that's usually something where if we can get really drill down to the details and assess specifically what the auditor's concerns are, maybe we can help write an appropriate response.
Yeah, maybe they could use monitoring, right?
Right.
That they could have some kind of monitoring in place, because there really isn't any scientific value in running a sterility test on product once in a while.
Perfect. Well, I think that we're right up against the hour. And I want to thank you, Jen and Bryce, for all of the insights and this great informative presentation. I'd like to thank you, the viewer, for attending this session. We hope that you found this to be a valuable experience. This session was recorded and will be archived on the Nelson Labs website in the next couple of days. And if you would like to go back to review any of its content, you may do so on the Nelson Labs website or in the Sotera Health Academy, as it will be archived there as well. Again, we thank you for your time today, and we hope you have a great rest of your day.